Hello, I am Rosalino Vázquez López. I research at the Universidad de Anáhuac, Mexico. My team and I appreciate the invitation to this second edition of COVID-19 International Congress. One of my line of research that I have developed with my friend, the pediatric gastroenterology, Dr. Christian Boyo, is the study associated between COVID-19 and microbiota and dysbiosis. It is a pleasure for us to share today the presentation entitled Implications of Intestinal Infection with SARS CoV-2 and its correlation with dysbiosis and the gut-lung axis. Thank you.
Thank you for the introduction, Dr. Rosalino. This work is entitled Implications of the Intestinal Infection by SARS-CoV-2 and its correlation with dysbiosis and gut-lung axis. My name is Nicole Ponce. My coworkers are Lisbeth Prudencio and Roberto Herrera. Okay, so microbes play a very important role in our bodies, especially in the immune response against sickness. One of my partners will explain with more detail later, but the respiratory and the gastrointestinal systems are connected by microbes. With this new pandemic that's going on, scientists knew that it was caused by a respiratory virus and started to do more research to discover why patients with respiratory infections have had gastrointestinal infections. In a study made on April 2020 by Elsevier, from 2 to 49.5% of the patients have had diarrhea, vomiting from 3.6 to 66.7%, nausea from 1 to 29.4%, abdominal pain from 2.2 to 6% of the patients, and gastrointestinal bleeding from 4 to 13.7% of the patients. They knew that the symptoms were caused by a dysbiosis in the gut. Later on, they started questioning how a respiratory virus could cause this gastrointestinal microbiota dysbiosis. Dysbiosis in the gut microbiota has a pivotal role in the immune response, but not only this dysbiosis can also create a chronic inflammatory bowel disease, Crohn's disease, and more diseases. This microbiota decreases within aging. This may be the cause of severity of the symptoms and the infection itself, hand by hand with the comorbidities of this infection, such as hypertension, diabetes, and obesity. The main symptoms of the infections are fever, cough, and fatigue. But a low number of the Wuhan patients have referred to have headache, diarrhea, vomiting, and abdominal pain as common symptoms too. However, as the pandemic advanced, more and more research was made worldwide, and gastrointestinal systems started to seem more and more common. Due to the common symptoms, the main affected organs with SARS-CoV-2 seems to be the lung. However, during the course of the disease, organ failure and dysfunction started to be common, such as the liver and intestine. With this information, scientists collected fecal samples and explored them. What they found was intriguing. They found SARS-CoV-2 nucleic acid in the fecal samples. Despite clearance of viral RNA in the respiratory systems, the fecal sample of patients infected remained positive for about 11 days after the sample was taken. Some of the previous studies from other patients infected with SARS also described gastrointestinal symptoms like diarrhea. In this past ratio between 16 and 73% of the patients, and now with the SARS-CoV-2, the ratio goes between 2 and 20%. The difference might seem a lot, but this difference might be due to the diagnosis criteria. We should not take for granted the fact that both respiratory infections we find respiratory and gastrointestinal symptoms, or even just gastrointestinal symptoms. Thank you.
We need to remember the COVID-19 or new coronavirus was detected for the first time in the city of Wuhan, China in November 2019. The mechanism of transmission of the disease is from person to person through the air, through fluid droplets that are exhaled when coughing, sneezing, or speaking, and are deposited on surface or directly on the host. The etiological agent that causes this disease is SARS CoV-2. It is classified as a respiratory virus. There are various factors that determine the pathogenesis of this RNA-type virus that, upon releasing its content in the host cells, initiate the replication producing the structural viral proteins as shown in this picture. However, the most important protein in this issue is the protein S or PIC. To the characteristic and classification of the virus, everyone could think that it will only cause pathogenesis in the respiratory system. However, it is known. It has been shown that this virus causes alteration at the intestinal level. The type 2 of angiotensin converting enzyme is a type 1 of membrane protein that under normal condition is widely expressed in the body, has receptor in the heart, kidney, lung specifically, and expresses in the epithelium of type 2 of pneumocytes, and the intestine where it expressed in the brush border of enterocyte, where the intestinal microbiota has a very important role. The type 2 angiotensin converting enzyme is an enzyme that has a trivalent function. It functions as a negative regulator of the renin-angiotensin-aldosterone axis as amino acid transporter, and currently the virus has been identified as SARS-CoV-2 receptor. The virus binds to this enzyme through protein S virus that has surface of the coronavirus begin a proteolytic modified, resulting in blocking its protective effect and increasing levels of type 1 angiotensin converting enzyme, leading to process of inflammation, vasoconstriction, fibrosis, and intestinal dysbiosis, as has been observed in patients with COVID-19.
Well, now I'm going to talk about the gut-lung axis as well as the microbiota and its correlation. Nowadays, we are aware that the human body is an organism that lives in constant symbiosis with a great variety of microorganisms that inhabit it and that are of great importance in maintaining our health. The human microbiota as a whole is made up of 10 times more cells than the human body itself. In fact, around 7,200,000 bacterial genes have been identified, of which almost half are present in the intestine, compared to approximately 20,000 coding genes and another approximately 20,000 human non-coding genes. Therefore, we could consider the human body as a large holobiont organism that is strongly influenced by the great diversity of microorganisms that inhabit it. With that being said, it is currently known that microorganisms that live naturally in certain specific regions of our body not only have their effect in the local area where they live, but also have a significant impact on other regions and systems of our body. Indeed, there is a communication already described between the intestine and the lung in such a way that one has an impact on the other. When we get sick, the intestinal microbiota plays a great role in the immunity of the lungs and the defenses of our body, involved with viral respiratory infections, as is the case with the SARS-CoV-2 virus. The lung microbial composition and immune responses are influenced by the intestinal microbiota through the direct setting of bacteria in the respiratory tract or through the distribution of bacterial metabolites such as short-chain fatty acids, which promote the growth of certain bacterial enterotypes, such as bacteroids, which are associated with a healthy intestinal microbiome, as well as participating in the biosynthesis of certain vitamins such as B2, B7, and C. And these metabolites can also act as immunomodulatory molecules and are therefore strongly involved in development of diseases. When the function and composition of the intestinal microbiota is affected, which is defined as dysbiosis, it can generate complications of the disease in question. The intestinal microbiome can be affected in various ways, such as diet, medication, and the development of lung diseases, which is associated with an alteration of the immune responses and the maintenance of homeostasis in the respiratory tract. Currently, it has been proven that there is a strong relationship between SARS-CoV-2 and intestinal microbiota dysbiosis, which makes the patient more prone to both pulmonary and gastrointestinal complications derived from COVID-19. Another way in which the gut microbiome may be affected is by being older. Elderly adults are inevitably more susceptible to developing respiratory diseases, but not only due to the aging process, but it is also related to changes in the intestinal microbiota. Dysbiosis due to the advanced age of this intestinal microenvironment may be responsible for generating the complications caused by SARS-CoV-2.
The human intestinal microbiota consists of more than a trillion microorganisms in a complex and dynamic ecosystem, regulating the immune system and our entire physiology. Firmicutes and Bacteroidetes are predominant in gut, while Proteobacteria, Bacteroidetes, and Firmicutes preponderate in the lung. These microbes play very important functions in the body, including nutritional metabolism, development and modulation of immunity, as well as defense against harmful pathogens. In the gastrointestinal tract, the epithelial barrier protects against the invasion of pathogenic microorganisms and helps keep intolerance to food antigens, while it may also be associated with systemic and pulmonary immune functions. Once damaged, microorganisms translocate into the bloodstream or lungs and can induce septicemia or acute respiratory distress syndrome. Complex interplay and alliance between immune system and gut microbiota regulates and support each other as 70 to 80% of the total body's immune cells that are present in the gut. Dysbiosis, defined as changes in gut microbiota leading to microbiome imbalance, has not only been closely linked with the pathogenesis of many inflammatory diseases, but plays a critical role in diverse infections as well. There is evidence for a crosstalk between the respiratory tract and the gastrointestinal tract, or more precisely, between the intestinal microbiota and the lungs, and this connection is named the gut-lung axis. Patients with COVID-19 suffer from fever, cough, myalgia, fatigue, and pneumonia, which might aggravate to acute respiratory distress syndrome or multi-organ dysfunction. Various studies have found the presence of gastrointestinal symptoms during the course of disease, which are nausea, vomiting, diarrhea, and abdominal pain. These symptoms might be either due to direct infection of the enterocytes by SARS-CoV-2 through a phenomenon of gut-lung axis involving the gut and the lung microbiome, or through immunoregulatory mechanism. Changes in the taxonomic composition and decreased diversity and function of the gut microbiota, known as dysbiosis, can affect the immunity of the lungs. On the other hand, the respiratory tract has its own microbiota, and lung inflammation can lead to intestinal dysbiosis as well. Recent studies hypothesize that endotoxin, microbiota metabolites, cytokines, and hormones from the gut could reach the bloodstream and the lung niche in a bidirectional gut-lung axis crosstalk. The host immune status is influenced by intestinal microbiota and may influence the extent of the immunity to viral infections, including the SARS-CoV-2. A number of COVID-19 patients had gastrointestinal symptoms and prolonged manifestation, mainly the diarrhea, that were inversely correlated to decreased microbiota richness and diversity associated with immune deregulation and delayed SARS-CoV dose clearance. It is speculated that the expression of ACE2 receptor, which is required by the SARS-CoV-2 to enter the host cells, might be modulated by the gut microbiome. Gu et al. In China evaluated the intestinal microbiota from 30 COVID-19 subjects, 24 H1N1 patients, and 30 healthy control. Subjects infected with SARS-CoV-2 had a decrease in the diversity of the intestinal microbiota when compared to controls, with predominance of opportunistic genera such as Actinomyces, Rothia, Streptococcus, and Veillonella, besides a decrease in the relative abundance of beneficial microbes such as Bifidobacterium genera. As dysbiosis might be an essential factor in disease severity during COVID-19, maintaining a healthy gut is essential during this pandemic. Diet with low fiber and high fat carbohydrate are usually responsible for gut dysbiosis, and this change in homeostasis might be responsible for an altered immune response. Healthy balanced diet, rich in cereals, whole grains, legumes, fruits, and vegetables is advised to COVID-19 patients who are asymptomatic or patients with mild symptoms or in quarantine. The inverse correlation between the consumption of dietary fiber and the serum levels of C-reactive protein, interleukin-6, interleukin-18, and tumor necrosis factor alpha, which are strong inflammatory cytokines, is the main reason behind emphasizing this kind of diet. There are a growing number of studies evaluating the effect of probiotic or prebiotic administration in reducing the incidence, duration, and severity of viral respiratory infections in humans. In view of the current knowledge, the modulation of microbiota is being investigated as a possible adjunctive therapy for COVID-19. The potential for probiotic use, that is a live microorganism that, when administered in adequate amounts, confer a healthy benefit on the host, is supported by experimental studies, meta-analysis, and clinical trials on influenza virus, rhinovirus, and respiratory syncytial virus. Although the mechanism have not been determined in SARS-CoV-2 infection, some probiotic strain present antiviral properties in other coronaviruses. In addition to prevent lower and upper respiratory infection, probiotics could assist in the treatment of diarrhea associated with SARS-CoV-2 infection itself or caused by the antibiotic used to treat secondary pulmonary infections. The improvement of the intestinal microecology and the eubiosis process by taking probiotic may promote a regulatory immune system and prevent an excessive inflammation or secondary infections. Some strains of Bifidobacterium, Lactobacillus paracasei, and Lactobacillus rhamnosus reduces the occurrence of respiratory infection, such as H1N1, H3N2, and H5N1 by boosting the vaccine immune responses. This improvement of the microbiota mucosal immune system interaction could also benefit the immune responses to vaccination against SARS-CoV-2 virus. Nevertheless, we hypothesized that dysbiosis of microbiota modulation could potentially affect the efficacy of COVID-19 vaccines. To date, there are no current published studies regarding the relationship between the gut-lung microbiota and vaccination to this infection. To conclude, healthy gut microbiota can control the lung infection caused by SARS-CoV-2 by producing large number of immune cells as compared to a smaller number of immune cells by dysbiosis of gut microbiota. Diet probiotics and prebiotics are the crucial modulators in regulating gut microbial environment and might be favorable in maintaining the homeostasis of gut microbiome and might affect the SARS-CoV-2 infection. The purpose of probiotics and other ways aiming to modulate the intestinal microbiota in COVID-19 demand more explorations, particularly randomized, double-blind, controlled clinical trials, including larger cohorts in different ages and disease causes.
Bon, merci pour votre attention. J'espère que cette vidéo vous a plu et que le sujet a été de votre plaisir. Des salutations du Mexique.